Yeah, so lot. So this is just a definition what this is. This is an electron transport chain. It's a series of membrane proteins. So it's a system, basically. There's a lot of proteins here. Electrons are jumping on these and then releasing energy. Like I said, we will cover it when we cover chapter 6. Photosynthesis, actually, we will cover it for chapter 5 too. Photosynthesis and cellular respiration, they both use electron transport chain for the release of energy. So they both use it. Okay, this is a review slide again. ATP, we know that it stands for adenosine triphosphate. It's the currency for energy. So when this phosphate bond breaks, it's going to release energy. There's potential energy here. It has stored energy. When it breaks, it's going to release energy. So tri here stands for 3 phosphate bonds, triphosphate. So once it loses, so this is the energy currency, and we learned last time that's made in mitochondria. All cells rely on potential energy stored in ATP to power the chemical reactions, power the metabolism that's going on. So this phosphate bond can break through hydrolysis. Hydro means water. Lysis means breakdown. Hydrolysis. So this chemical bond breaks with, once it comes in contact with water, this breaks, it's going to release energy. This reaction is called hydrolysis. Once that happens, you're left with this here. You're left with 2 phosphates and this molecule here. Now this is not called ATP, it's called ADP, and that stands for adenosine diphosphate. Because you have 2 phosphates, di means 2, tri means 3, you have 3 phosphates here, so that's called ATP. So removing the phosphate group is going to release some potential energy that's stored in ATP, and the cells can use that energy to do metabolism, to do work, to move maybe muscles. So that energy can be used for all those. So let's see. So I said ATP has 3 phosphate bonds. Here, I want you to think of it as maybe, your cell phone battery, it's full here. And comes in, reacts with water. So you eat some food and you get energy from that, and you break it down through hydrolysis. That's going to give you energy to do work. Now you're left with ADP, adenosine diphosphate, and broken phosphate bond. So this needs to replenish. This is called ATP synthesis. This happens through an enzyme that's called ATP synthase. ASE ending is for enzyme, so ATP synthase. So that's going to replenish. So this is not full battery. This is half battery. You started using it, it went down because you only have 2 phosphate bonds. This is going to replenish that. So when you eat food, that happens. It works with the ATP synthase, and you have full ATP here, full battery again. And again, with water hydrolysis, you can release energy again. So that's basically the cycle. ATP synthesis is coupled with energy releasing reactions. Hydrolysis is coupled with energy requiring reactions. Because after hydrolysis, it's ADP that's left. Now you need some energy to basically replenish this to make it full again. So ATP, it's going to provide energy. So I said that the hydrolysis, it requires energy. So it's going to provide energy to build larger molecules, and we call them polysaccharides. You have a glucose molecule here. You want to take this glucose molecule, you want to put it on this chain here. This is a short polysaccharide. You need energy for that. So once, it takes the phosphate group, it activates this, and it's going to go basically connect with this chain. Now you have a longer polysaccharides. So basically, you need energy to build the larger molecules out of small subunits. If you have a lot of glucose in your body, your liver wants to save it, and it wants to put it, convert into a longer polysaccharides like glycogen. It's going to use energy, do that, and then store it. And then this phosphate group, this activated phosphate group, it reacts with this short polysaccharide, and that's how it makes it longer polysaccharide basically. So ATP donates a phosphate group that changes the shape of the target molecule. So what does this mean? Last time we looked at the plasma membrane. So I want you to pay attention here. You see the hydrophilic heads, and then you see the hydrophobic tails. If you remember, I said this phospholipid bilayer creates a different environment inside the cell versus outside the cell. It's going to let some things pass through, but it's not going to let molecules pass through that are maybe big in size, or maybe they have a charge or they're polar. So it's going to need a channel. It's going to need a transport protein. And we defined last time transport protein as something that's going to take things from one side and put it in the other side. So that these are transport proteins here. Some of these channels, they open when they're activated basically, and you need energy for that. So ATP donates a phosphate group that's going to change the shape. So what you see on this photo, you have ATP that turned into ADP. That means one phosphate bond broke attached to this protein here, and it caused it to open. So now these things can go to this side. So some of the transport proteins, they require energy. So this is just to tell you that enzymes, they speed up reactions. So chemical reactions, they need to happen very, very quickly, and we have enzymes for that. So I said that enzymes, they speed up chemical reactions. You have enzyme salivary amylase in your saliva. It's going to help break down starch very quickly, versus if you didn't have it, it will take forever. So they must happen quickly, and you have enzymes for that. So this is, again, should be reviewed because I sent out a video as well. So enzyme is a protein that's going to speed up a chemical reaction without being consumed. There's nothing happening to the enzyme in this whole reaction. So you have the substrate that needs to be broken down into smaller pieces maybe. You have the enzyme. This substrate has a shape that needs to fit with this shape here, and that's called active site. This is called active site. This needs to fit here. Once that happens, we call it enzyme substrate complex, and it's going to break it down. Now another substrate can come in, attach here, can be broken down to products. Another one can come in, same thing. So all I'm trying to say is there's nothing happening to the enzyme here. So there's nothing happening to the enzyme in this whole process. This is just one example. So this is just one example where the substrate is being broken down into products. There are enzymes that build things. So you might receive substrates that look like this. It goes through the process, and it's going to be, this is going to be product. So it can go this way or it can go this way. So substrate molecule binds to the active site. That's where the reaction happens. And then once the chemical reaction happens, the product molecules are released, and then enzyme goes back to the original form. So not all enzymes break a single substrate into two products. Other enzymes combine two substrates into one product. So it can go this way. How do enzymes speed up chemical reactions? They do it by lowering the activation energy. If you're taking chemistry, you probably already know this, but if not, I want you to remember maybe this definition. If I ask you on the exam, how do they speed up the reaction? You're going to tell me it happens because the activation energy gets lowered. I'm not going to go in more detail. That's all I want you to know. They speed up reactions by lowering the activation energy. In this photo, this is a reaction happening where there's no enzyme present. Then you have an enzyme, it lowered the energy that was needed for the reaction and also sped it up. So it speeds up the reaction and lowers the activation energy. So the reaction happens faster because there's less energy needed. So this is just to show you that sometimes the energy it's going in if it's building, and sometimes energy is coming out if it's breaking it down. So if you're eating food, it's getting broken down, maybe there's energy that's coming out. If you're trying to store something in the liver, then energy might be needed to make it a longer polysaccharide and then store it in the liver. So how does the cell control the rate of chemical reactions? How do you know that you have enough of this product to work anymore? You want the enzymes to stop working or they're doing the reaction. So how does that happen? So actually, before I go into that, I want to know if anybody is lactose intolerant. Anyone lactose intolerant?
Yes.
So what happens when you eat something with lactose? Probably gives you upset stomach.
Like an acidic reaction, scientifically speaking.
Okay. So lactose intolerant enzyme. So this is what's happening in your body. Your body has an enzyme called lactase. ASE ending, it's an enzyme. OSE, it's a sugar. Lactose has the shape. So lactose has this shape. It's going to fit with this lactase, and it's going to break it down into galactose and glucose. If you have a DNA mutation, your body doesn't make lactase, or it doesn't have lactase, or if you have lactase, it's not able to attach it, then you're going to be lactose intolerant. Another thing I want you to notice from this photo, here, that's the shape that's going to fit with this enzyme. Here is a sucrose, that's another sugar. It has a different shape. It's not gonna fit with this enzyme because it has a different shape. So each type has its own enzyme. Sucrose enzyme is probably called sucrase, ASE ending. Lactose, lactase. So shape is very important. And if you're lactose intolerant, sometimes people think they're lactose intolerant, but they're not really lactose intolerant, and it might be because they're not drinking milk or maybe eating milk-related foods that often. If they don't do it very often, then sometimes they also get upset stomach. So if you don't have that enzyme, so if you don't have that enzyme, see, then this might be a little bit hard to see. I'm trying to find another photo. Okay, here. So if you don't have this enzyme lactase, then there's bacteria that's found in your gut. And that's the bacteria that's gonna try to break down that lactose. When that happens, it's gonna produce as a waste product a lot of gas and some acids, and that's what gives you the upset stomach. So that's why you get cramps after if you don't have this enzyme. So how you can prevent that? Maybe before you eat something with lactose, you can take this digestive enzyme that's lactase so it can be broken down. So you can avoid that by taking these. Also, they put there's this yogurt, they take out all the bad bacteria. So if you were to drink raw milk, it's going to have a lot of bacteria in it. In this yogurt, they basically take out the bad bacteria, or in these types of yogurts, they put good bacteria. They add good bacteria to it. So if you maybe, eat this yogurt, it's going to give you a good bacteria that's going to help you break down things. So they do that. And that's why they call it probiotic. So that's why it's called probiotic. Another place you might see this is so kimchi. What they do, they take cabbage and then they put some oysters in it and then they let it rot. The rotting is the bacteria. So if you eat that, it's gonna have a lot of bacteria and it's supposed to be good bacteria. So it's gonna give you good digestive health. So that kimchi and also miso. Miso. So these are the soya beans. They let it ferment for a while. And when that happens, obviously there's bacteria that's breaking it down. If you eat that, that's gonna give you good digestive health as well. Have you guys heard about fecal transplant? No. So basically, if someone has lost all their good bacteria from their digestive tract, they take some poop and they put it through your throat into your digestive system to give you that good bacteria. So there are a few papers on it. If you're interested, you can search it. It's called fecal transplant. So that's what they do to give you some good bacteria there.